welcome everybody to this evening and this morning's seminar, and it gives me great pleasure to introduce Dr. Marta Peirats-Llobet, who's going to be giving us the presentation today. I've had the pleasure of interacting with Marta on a number of occasions in a host organisation, which is the Trobe University, but also at a couple of conferences. And she's been doing some really interesting work in barley, particularly around barley germination and spatial transcriptomics. So I think it's a great fit for this audience, and I'm really looking forward to her presentation. So Marta is actually from Spain, so she completed a PhD in biotech there. And in 2018, she moved to Australia, which was obviously an easy choice, given Australia is such a great place to live. And she took up a postdoc position at the Trobe University, working with Professor Mathew Lewsey, who himself is quite renowned for a number of genomic techniques, and later moved to Jim Whelan's lab. Jim is no longer in La Trobe, he's based in China, but Marta's still in La Trobe doing some great work where she's optimizing transcriptomic technologies in a number of plant species. So she's used laser capture, single cell profiling, and I think probably more relevant to the talk today is the spatial transcriptomics. So really excited that she's been willing to give the presentation. And just to note everybody that this is being recorded, you got that message. She's going to talk for around half an hour. If you've got any questions, please post them in the chat. And if not, if you've got other questions, we're happy to take them live in the session, and I'll help to facilitate that later. So I hope everyone can see the screen, and please, Marta, take it away.
Lovely. Thank you very much, Matt. If everybody can see my screen, I will just start. So first of all, thank you very much for inviting me to share our work with the Barley Hub. My name is Marta, and I'm a plant molecular biologist that happens to work on barley. This is a technology-driven talk that aims to provide the community with a state-of-the-art technology that will allow us to visualize barley gene expression across germination in time and space. Let me see if I can, seeds make up a large proportion, around 70% of both animal and human diets. And needless to say that many commercial interests of grains. Since barley is mainly used as a grain, control of germination has been one of the most studied processes. However, classic genetic studies of germination have their limitations in understanding tissue complexity. So how do we study tissue complexity? Let's imagine our complex tissue represented here as a fruit ball. If we do some classic bulk RNA-seq, represented as this fruit smoothie, you will have your sample blend and you will obtain gene expression, but it will be just a mash. You could get a bit more precise and do population enrichment, so you will separate some of your tissues and then do bulk RNA-seq. But you could do a bit more. You could separate individual cells, barcode them, and then sequence. This would be what a single cell experiment looks like and could be compared to a fruit salad. Within that fruit salad, you will know the components of the fruit ball, but they will be, the location on the fruit ball will be lost. So to fill up that next level of location, there has been this new technology called spatial transcriptomics. In this case, spatial transcriptomics will give you gene expression of your tissue without actually losing where everything is located. And we thought that this would be a good approach to study a complex process like is germination and a complex tissue like is the barley grape. So how does Visium work? When I started this work in 2020, few technologies were available and none of them were specifically for plants. So we selected Visium, which is the spatial solution from the Next Genomics. This solution is a slide-based gene expression approach that uses this glass slide that contain 4 capture areas that look like this. These capture areas are coated with these barcoded spots. They have around 5,000 in each capture area. The barcoded spots are 100 microns apart, center to center, and each of the spots have 55 microns in diameter. So that is the resolution of the spatial transcriptomic video. These barcoded spots contain these barcoded oligos that you can use to track your mRNAs. For example, you will bind this region, poly-DT, to the poly-A mRNAs. You will be able to follow each of your mRNAs with a unique molecular identifier, and those mRNAs could be traced back to their spatial location on the spots because each of the spots will have a spatial barcode. So Visium uses 2 different workflows or kits. The one at the top is called tissue optimization, and this is used to help you identify how long do you need to permeabilize your tissue to release the mRNAs on top of the microarray. This is specific for each tissue, so you will need to optimize that time for the different tissues. And the second kit is called the gene expression kit, and these slides will contain those spatially barcoded oligos that I just mentioned. Since these kits were designed and optimized for mammalian tissues, I had to make it work for plants. And doing so was especially difficult for barley grain because of the amount of starch and how hard the grains were. So how was the tissue optimization? You can see here the current protocol from the 10x website for mammalians. So some of my optimizations are shown in here in all these colors. For example, some of the optimizations I did for barley included cryosectioning. So you needed to optimize the material preparation, how you freeze your samples, the temperature of the cryostat. You need even to decide the hardness of the blades that you need, thickness of your tissue, and even how to place. You need to learn how to place the sections on the slide. Then I had also some problems with my samples getting, they were not intact because the stain, which is recommended, toluidine blue, was dissolving the starch. I also tried a couple of permeabilization solutions, and lastly, the tissue removal was a very important step since we needed to optimize for barley. As you can imagine, mammalian tissues don't have to worry about cell walls, but we had to select at least a good combination of enzymes that allowed us to remove that tissue. Then you will just synthesize cDNA of your mRNAs, and then the oligos will have fluorescent tag that you could follow the footprint of your sections. Once I was happy with the optimizations, I followed through to the gene expression slides. So a quick overview of the experimental design is shown in here. We selected 5 time points across the 24 hours that takes barley to germinate. For each time point, we produced 4 sections from the same seed. So we will have replicates. This protocol requires, same as I showed you, preparing your cryosections. This is how one of the sections look like. And then you need to fix, stain, and image your samples. This is actually the imaging the bright field image of one of my tissue sections. The next step will be to apply the permeabilization time that you already optimized, and you will release the mRNAs on top of the microarray. Then the cDNA will be synthesized, so you will have your mRNAs attached to the spatial barcoded oligos. Then you will just have to create your barcoded libraries that will be sequenced using an Illumina platform, and then you will do your spatial analysis. So after the data was analyzed, this is a representation of one of the sections that I just showed you. For example, this represents 24 hours. This is a dimension reduction plot called UMAP that shows you the clustering, and the dots in this plot correspond to the spots on the slide, and they are grouped using a similar gene expression. That's done automatically via software. And for example, you might have seen these plots for single cell. They are the same kind of plot, but in single cell, every single dot of this plot corresponds to a single cell. In this case, because spatial transcriptomics at this point is not it doesn't have single cell resolution, each of the spots here corresponds to a few cells with that being said. So you can see the different colors representing clusters. But because we can trace back where these spots were on the section, we just overlap them onto our bright field image. And as you can see, spatial transcriptomics recapitulated perfectly the tissues that you would expect in a barley grain. We were very excited at this point, and since this was the very first time that this was done on barley, we needed to validate our data. Firstly, we followed the informatic marker gene identification, and this plot here shows you the markers that are genes that are highly expressed and specifically expressed within the clusters. For example, if you look at the coleorhiza row here, these 4 genes are highly expressed. You can see the expression in here, so they are blue, and you cannot find them in any other cluster. So once we were happy with these markers, we wanted to actually see them or validate it using a more classic technique, which is in situ hybridization. So we pick one of the coleorhiza genes. You will see this is a thionine that is a coleorhiza marker, and you can see how the expression of thionine is actually in the coleorhiza. But then we wanted to see how the expression of this gene looks using spatial transcriptomics. So this is our sample with the thionine expression in the coleorhiza. It's important to mention here that spatial transcriptomics is a quantitative technique. So you can see that the different dots have different or a gradient of colors, meaning that the expression of the thionine is actually very heterogeneous. So now that we were happy that the results made sense, we analyzed all the samples and validate them, and then we were ready to dive into the germination process. The graph that you see here illustrates the processes happening during barley germination as imbibition happens. In phase 1, you have rapid water absorption. When this water absorption plateaus, you will find yourself in phase 2, where the metabolism reactivates. There is reserve mobilization, fermentation, and some other processes. And that reserve mobilization will produce the energy necessary for the plant to move to the next phase, which is growth. And for example, at 24 hours, when the radical emerges, that will be germination sensu stricto considered finished. So let's see how the spatial data illustrates this. To have a bigger picture of what was happening in germination, we generated this type of bubble plots using gene expression data, spatial gene expression data, and a list of annotated genes. For example, this plot shows you in a gradient of colors the gene expression. So the more blue the dots are, the highest the expression. And it's important also to see the size of the dots because they will represent how much of that gene is expressed in that cluster. For example, if you have 20%, if you have this size, will mean that 20% of your spots contain that specific gene. So 1 out of 4 will have that gene expression. These plots are a bit more dense, so there's more information contained in the plot, and you can see what is expressed in this axis. So these are all genes. Then when are these genes expressed? Because we have done a time course and because we have done a spatial transcriptomics, you can follow the tissue that these specific genes were expressed in. So I will walk you through this specific bubble plot now, which is representing transport-related genes. It's well known that we all know that transport of water, iron, solutes, and other elements is essential for successful germination. So we selected these genes, this gene list, and I'm just going to zoom in a specific group, which are the aquaporins, which are membrane proteins that facilitate transport of several molecules. If we zoom in a little bit more, you can see that the tips, tip 1,1, for example, this aquaporin, has a specific gene expression in this tissue here. So the blue dot corresponds to the coleorhiza. And this can be seen across the early time points of germination. And you can see the expression in coleorhiza. And suddenly at 24 hours, that expression expands, and you can see tip 1,1 expressed in the embryo tissue, so coleorhiza and mesocotyl. For example, another aquaporin has a totally different expression pattern. Aquaporin 3.1 can be seen already in all the embryo tissues in early development, all of the time points, so here. And then at 3 hours, you can see that they are expressed in a different cluster, which is, in this case, the scutellum and aleuron. You can actually see the gene expression here. And interestingly, at 24 hours, there's not much expression anywhere but in the aleuron, and that's exactly what you can see here. Another important transport-related gene is the proton ATPase. So this proton pump moves protons to acidify the apoplast of the cells where this gene is expressed. The proton pump, you can see localized or expressed in the aleuron tissues at most of the time points, and at 24 hours, that expression is expanded to this area adjacent to the scutellum and also across the aleuron. The function of this proton pump in aleuron and endosperm will be to acidify the apoplast to generate a perfect environment for the digestive enzymes that work in those areas. Talking about starch, we all know that barley, as many other cereals, store their energy reserves in the form of starch in the endosperm, which will be degraded during germination to ensure embryo growth. So here you have plotted a list of starch metabolism genes. And if we focus in one gene, this beta amylase, that degrades starch into smaller oligosaccharides, is highly expressed across all the endosperm tissues all along the full germination. If you look at some other amylases, in this case alpha amylases, that also degrade starch, but you can find no expression of these three genes in the early germination, and you find it concentrated at 24 hours in a very specific region. You can see the three different genes have expression in this endosperm region closer, so in the junction of embryo and endosperm. And this is how biologists, people that work on barley, can use the spatial transcriptomics to navigate or investigate complex processes. Another example of the potential of spatial transcriptomics is this co-expression analysis of spatially variable genes. We focus here only on embryo tissues. So spatially variable genes are genes that have a pattern in space that is not random. So for example, we found that 24 genes, auxin-related genes, had this spatially variable expression, finding 18 of those expressed at 24 hours. And from those 18 genes, 7 were involved in transport of auxins. So these 7 genes expression patterns are shown in here. You can see that the patterns are complementary, suggesting that transport and accumulation of auxins in the barley embryo during germination has a spatially complex pattern or distribution. This is actually well known for Arabidopsis, where there is a well-defined role of auxins in the formation of the apical basal embryonic axis during embryo development. As I showed you, spatial transcriptomics generates a huge amount of data, so we compiled it and make it accessible in this spatial transcriptomics explorer. You can check your favorite gene, snapping the red QR code, and you can check your favorite gene. So this is the end of the germination work that I've been doing. And during the last year, I worked on expanding this project that I just talked to you about. We expanded these longitudinal sections of barley with some other longitudinal sections during development. And we also did some more spatial transcriptomics on matching time points for germination, but in this case, they were transverse sections. So the main goal of this project was to integrate the 3 data sets and establish a barley grain atlas from after pollination to the end of germination. Since these are crucial stages on grains, we think that this atlas can actually help the community to identify genes with agricultural importance. You can pick genes and analyze grain size, dormancy, starch accumulation, whatever you can think of, and see their expression on the space. So after the germination work, I got the idea that we could be visualizing the gene expression or the spatial gene expression, adding a 4th dimension. This is the experimental design that made it possible. On each of the slides, I would prepare, the samples will be prepared as you can see here. I will cut my samples this way, and then I will move this little bit to the side. You will prepare the blocks with this, and then you will do the sections. These sections will be serial sections that you will place on the slide. And if you reconstruct those sections back to the original order, you will get this Z stack. So you will have the X and Y dimensions, the 2 dimensions, and then you will have a third dimension with the axis, so you will have a volume. And then you will get, because we have done a time course analysis, you will have 4 dimensions. And I don't want to go into many more details of the results that we have done, because we are still analyzing and finalizing these data, but I want to get a bit deeper into these bubble plots, because they really contain a lot of very useful information. I showed you how they work, but what I didn't mention is that, for example, this bubble plot contains 36 genes. There are also 9 time points, so 36 times 9, and then because we have done 4 replicates per sample, this would be, so this bubble plot will be equivalent to 1,300 in situ hybridization. What are the chances that you get 1,300 in situs, that they all work? We all know that in situs are not the easiest thing to do. But here you can see all the expression of all those genes without losing spatial context, and actually you can quantify those data points. And I will just finally show you a couple of examples. If we focus on 2 specific genes here, this granule-bound starch synthesis, they are the enzymes that convert linear dextrins into amylose, the ramification. You can see that, for example, 1A is highly expressed, or you can find it highly expressed in subaleuron. And this is how the reconstructed 3D sections look like with on this granule-bound synthase A. How cool it looks, the subaleuron tissue. And then if you look at 1B, the expression is totally complementary. You will see expression on the embryo tissues, and if you even look more in detail, you can see that the expression is highest in the plumule. So these 2 genes show you how a starch is synthesized within the space. And just to finish, I just wanted to show you that there are very interesting expression patterns that we could be checking on space. And sorry, it might be a bit too much going on, but you can see how different the expression patterns are in all these different genes that you can see here. And with that, I just want to thank the people that work in this actual work from La Trobe, our colleagues in Adelaide, and our collaborators in Sheffield, Hallam. And just some housekeeping, you can find the QR code to go to our database. And then if you want to read more about the germination paper, you can just snap this blue QR code. Lastly, a big shout out to the people at 10X that really helped us with the optimization. And just to finish with a fun note, I was really happy when one of my images made it to the cover of Nucleic Acid Research. And just now, just to finish, I will leave you with this quote from Sidney Brenner that talks about the importance of new techniques to propel science. And that would be it. I'm happy to take questions. Thank you.
Thank you very much, Marta. That was a fantastic amount of data and a fantastic number of in situ hybridizations. I was trying to think how many I've done through my career, and I reckon I'm probably up to about 200 or 300, but most of them have failed. So yeah, you've certainly.
That's what I mentioned. Yeah.
Yeah. Look, there's a lot of data there, and I appreciate people who take time to process. If there's any questions, I'm very happy to see them populated in the chat. I'm also very happy to take them live. I wonder, while I'm waiting for that to happen, if I might ask a question about the actual technique itself, which one of the steps you identified as being really critical is that tissue permeabilization step.
Yep.
So could you just explain, once you've established that permeabilization step for barley grain, have you been able to use the same conditions for all stages, or have you had to customize that for each stage?
Yeah, I think most of the tissues were the same. I have to mention, so I optimized also for the development data set, and since the samples are not that hard, because you can see the dry seed will be harder to permeabilize, the times could be a bit different. But at the end, for barley, there was not that much of a difference.
Okay, interesting. Yeah, because certainly those late stages, I guess if you think of the amount of starch and the quite prominent cell walls compared to some of the earlier stages where there's not quite as much deposition of perhaps thick cell walls and there's little starch, but it's good to know that can be used for different stages.
Yeah.
If you have a question, perhaps you can raise your hand. Otherwise, I will keep going. I've got another question that relates again to the technique rather than the biology. So you rely on messenger RNAs effectively adhering to the probes on the array. Do you have any idea how long the sequence is? I think you would be available. I'm not sure whether you've done it, just to get an indication of how much of the mRNA is actually linked to the barcode during the sequencing. Is it a base pair?
Yeah, I don't remember the exact number, but it's a fair bit. So I don't know. It's less than 100 nucleotides, I would say.
Okay, yeah. So it's relatively small. So when it comes to things like potential splice variants and the like, it would not necessarily be that straightforward to find them, particularly if it's towards the 5' end of the gene.
Yeah, probably. You will need to maybe design something different. Yeah, something different.
Yeah. And I'll keep going. So I think the particular cultivar you were using here was La Trobe, which is a really important Australian cultivar. But when using a reference for the spatial work, were you using the Golden Promise or the Morex reference, or a different reference again?
I don't remember I know it's like the genome version 51, if I'm not wrong, but I can't tell you on the top of my head what the cultivar is, but definitely it's not La Trobe. So we had some, I wouldn't say problems, but definitely the alignment of our samples to the genome, reference genome, wasn't great. So I think it was about 60%. So
maybe as the pangenome becomes more accessible to researchers in terms of utilization, it could be worth actually repeating. I mean,
definitely.
Not to repeat it, but
definitely. Yeah.
It said that it looks like the Morex reference. I'm pretty sure it is the Morex reference as well. But Peter's actually come up with an interesting question here, is talking about gene expression at 0 hours after, well, I guess it's, is it in inhibition? Yeah. So, and wondering whether that might be deposition of mRNA from previous stages of development. Is that, is it de novo sort of expression, or is it stored mRNA? What do you think?
That's what we think, that these mRNAs that you can see at 0, they have been previously synthesized during development, and they are just stored. Because as you could imagine, the seed at 0, when the moment it feels that there is water, things have to start happening. So at 0, you don't have the time, or the seed doesn't have the time to be unpacking the DNA and prepping the mRNA. So you just need some things prepared. And that is what we think is happening here, because we definitely can find some gene expression or some mRNAs at 0.
And so in terms of just following up on that, when you look at the grain, like I guess the post-germination set versus the grain development set, do you see more representation? Well, what proportion of the transcriptome do you actually see being expressed in, is it, I assume it's higher pre-germination, is that correct?
Definitely. So development has so much more expression compared to germination. Germination, you can see, as you could see in one of the graphs, at 24 hours, there's way more going on.
Yeah.
Early time points, it was a bit less, but compared to, it's like top and bottom if you compare development and germination. Yeah, way more in development.
So no doubt it will be fascinating to look at some of those genes and look at mutant phenotypes to understand whether they're actually doing anything during germination. It would be very interesting. Excellent. Are there any more questions online? I see there's some few comments. Isabel's asked a question here, congratulating you in the talk. Seeds have a large surface. Would this technique manage to get spatial expression at single cells of 10 to 30 microns, or would it include a few cells in each spot? I think that's a good question as well. Yep.
Yeah, so the resolution of this version of Visium, which is version 1, is 55 microns. So with that 55 microns, you will find a few cells. It could be from 3 to 10, they usually say. So at the moment, there is no technology available for us, or no Visium available for plants that you have a cellular or subcellular resolution. But they have been working on this new Visium HD that they claim they have a 5 micron resolution. So that would definitely be a way to go if you really want to go to single level resolution.
Just to add to that, and there are obviously competing technologies out there, and stomics, I think, is, or stereomics, or whatever it's referred to as, is a competing technique that professes to offer that single cell resolution, but a lot of that's kind of imputed from the analysis. It's not actually, not to say it's a bad thing. Certainly, it's an interesting technique, but I don't think we're quite there in plants yet for that single cell resolution. I see, hello Rungshuan, I see your faces appear, but there's also a couple of other people that have just popped a question in. Pete wants to know about the read depth for the sequencing. I wonder if you can recall what sort of depth you were looking at.
I'm terrible with numbers. I don't want to say something and then it's wrong, but I would say you need 50,000 double-ended reads per sample. Is that? Do you recall that, Matt?
I can't actually remember the exact amount as either. Certainly, we were sequencing it with, in terms of what we sent for sequencing for our similar experiments, we were doing more depth than we would do for a typical transcriptome. But yeah, it's certainly that the number of transcripts we were finding in young, fresh tissue is a lot more than what you would see in a grain. So, yeah, I can't actually answer that one directly, but perhaps it's something we can follow up with, Pete.
Yeah, definitely.
Eva's asked a question, then I'll get to you, Rungshuan. Sorry.
No worries.
Histological image of the 24-hour germination stage. There were some areas showing a lack of tissue, but there were spots that's still been assigned to clusters, potentially an orange cluster in the coleorhiza. Could this impact the accuracy of the spatial transcriptomic data? shouldn't these spots be removed from the analysis since there's no tissue there? I think that's a good question.
That's a definitely good question. The problem is that it could be, so you could find gene expression there, because probably when I actually put the tissue in, so the tissue was actually there. But you know that then you have to stain and image the tissue. So with the staining, you have to dunk the slide with some solvent, and you can definitely lose part of the tissue while you are doing that, but the mRNAs or some leftover tissues could be there. So when you take the image, the actual, it's not 100% exactly what you put in, because you manipulate the slide all the way through. So you are right that it might be more correct removing those spots, but I guess we didn't do it that specifically because this was the very first time that we were doing, and maybe there were some things that we could do better now if we do the analysis. So you are totally, yeah, you are right in there.
And perhaps just to add also, Marta, it is possible to view the data without the tissue section there at all. You can simply just look at the dots and just say, well, these are the dots. So yeah, the image can actually trick you into thinking that there's no image there when actually there no tissue there, when in actual fact there was tissue there when you initiated the experiment. So yeah, I think it's a good point though. Okay, Rung Xuan. Hello.
Oh, hey. You hear me okay? Right.
Yeah.
I just want to follow on the question that Matthew asked early on about, those sequences doesn't capture the 5' end of the mRNAs. I'm a bit confused about that because there's a lot of discussions, people saying different things. So when I look at the protocol, so after the mRNA being captured by the probe, you actually go through cDNA synthesis. That should go all the way to the 5', right? Ideally. So
yes.
When you do the sequencing, you're actually sequencing the whole of the mRNA. So is that true? Really, it doesn't capture 5' end at all, or it's just problem with the cDNA synthesis?
Yeah, that I could get back to you on that, because the thing
right.
is that you don't really sequence the whole thing, but you do generate that full mRNA, I would say. Or you
my understanding of the technology would be the mRNAs are pulled down by the probes. Then you do the cDNA synthesis. You actually have the whole of the mRNA. You probably chop them into pieces, like when you do the sequencing, like short read sequencing. I would expect that you actually capture the whole of the mRNA. But of course, I heard from conferences, people say, the technology, single cell technology 10x only capture the 3' end. I would expect there could be a bias somewhere, but I just want to understand a bit more about the limitation of this technology. That's my question. Sorry.
No, totally fair point. I could get back to you on that, because definitely you amplify the whole mRNA with your oligo. But then I don't remember exactly what happens after. How much do you sequence actually? After.
Right.
So I'll get back to you with that.
Okay.
Yeah.
Thank you.
Thank you for your question.
That's me.
Thanks, Rung Xuan. Sarah, who is online, she may still be here. She may have disappeared to go and deal with a trick-or-treater, but she was very happy with the talk as well. And she was curious to understand your thoughts on integrating spatial transcriptomics with also proteomics and metabolomics.
That would be actually the coolest thing ever, because I love this idea of that you can see. I'm a very visual person, and I think seeing the transcriptomics, metabolomics, and proteomics all in the same sample, that would be the ultimate goal, right? We want to see that all at the same time in the same sample. And I have seen maybe one paper that they actually do on the same Visium slide, spatial and metabolomics. So I always wanted to try, but didn't get the money nor the time to do it. But that is actually possible. It was done in a brain, as everything else, mouse brain, but that's definitely a possibility. Yeah, that would be really cool.
And there's a question for Mika, and thanking you again for the talk, but very curious to know what number of genes are you seeing in each spot? So that's another good question.
Yeah, that's again, can't remember names because it depends. It depends. For example, they do an averaging of the gene expression per spot, and depending on if you are doing a development, the number is really high. And if you are doing germination, that average is lower. I think we could be talking about 1,000 genes per spot during development, if I'm not mistaken, and around the hundreds during germination. So yeah.
Excellent. Look, we've had a good number of questions there. Sarah, your mic is popping up. I'm not sure whether you would like to ask a question or if it's just.
No. I was just going to thank Marta, but I've done that in a text. Sorry, I'm doing an enormous amount of juggling with a very hyped up little zombie who's just eaten a lot of Australian sweets. So, but that was a fantastic talk. Thank you so much. Yeah,
Yes,
that was super interesting.
Thank you very much. That was
okay, Tom. Bye.
Excellent. Well, I think I will just hold off for another minute or so and see if there's any more questions. But so Marta, I guess the intention, like you said, is you've made the germination data available in that database, and the intention is to incorporate that grain development series as well. When are you thinking that might be available for the community so they can have a good look at it?
We would want to do it as soon as possible. We are kind of finalizing a first draft that, we might knock to your door soon to see if you want to take a look. But we are planning, if everything goes well, to submit early end of this year or early next year.
Fantastic. I'm sure that will generate a lot of interest, and everyone will look at their favorite genes and expression patterns, and it'll save them doing 1,000 in situs to check every gene. So that's great. Look, I'm happy to call it to a close there. I think it's been a really interesting presentation. There's been some great questions and discussion, and Robbie apologised to me earlier for not being able to attend, but said he'd certainly make the effort with a number of other colleagues to have a look and then find some more questions for you to flick your way via email. So, Marta, thank you very much for taking the time late in the evening here in Australia to give that presentation. And thank you everyone for taking the time to attend the seminar. And I look forward to seeing you again soon.
Thank you very much, everybody. Thank you. Bye-bye.
Ciao.